The first part of the plant that we're going to go into detail about in this chapter is the roots. And that's not a bad place to start. You know, you start at the bottom and you work your way up, right? When we think about the functions of the roots, there are basically three functions that all roots are going to serve. First of all, they're going to absorb the water and the nutrients. So that's something that you probably already knew, but there are some people who thought that if you water a plant and you get it all over the leaves, that the leaves can absorb some of the water too, but that's actually not true. The water absorption has to happen through the roots and then through the soil. Okay, so it's going to absorb water, but also some of the minerals and things that the plant will need as well. So that's done through the roots. The roots are also going to anchor the plant to keep it in one spot. And they're also going to store food. Well, I mentioned carrots in the last video. Carrots are a really good thought about how plants are going to store food. The carrot is a really good example of that. All right, now, if I take a root and I slice it lengthwise down the middle, I get something that kind of looks like this. And you do have these diagrams in your book, but in order for me to be able to point things out to you, I do have them drawn up here. The book is a much better picture than I am an artist, so I just want to point that out. I'm not going to try to make you think that my drawings are better than what you're going to find in the book, because they're not, but I am able to point things out a little bit better this way. Okay, at the very tip of the root, you have your root cap, and the root cap is basically full of dead cells that are going to protect the root as it pushes its way through the soil. It's got to exert a lot of pressure to be able to push its way through hard soil, and so the root cap is there for protection. Okay, behind that you've got your meristematic region, and you will remember that meristematic and mitosis begin with the same letter. They begin with M, and so this is where the growth is centered in the root. The apical meristem, right down here, right by the root cap, and right centered in the center of the root, that's where most of the growth is concentrated. That's the absolute center of growth for the root. And then as the root continues to grow, then eventually these cells that are formed and down here in the meristematic region will start to elongate, start to become longer, and then by the time they get up here, they are becoming mature cells. Okay, so these are the four main sections of the root, and that's if I slice it long ways. But I'm going to be able to learn a lot about a root if I slice it crosswise. Now, the pictures in your book, and they're on the bottom of page 483, we're going to be doing a lot of comparison of monocots and dicots in this chapter. You will recall from chapter 11 that monocot, which stands for monocotyledon, and dicot, which is short for dicotyledon, has to do with how the seeds are formed and whether seeds have just one cotyledon or whether they have two. And there's vast differences in the plants themselves, in their roots, in their stems, and their leaves, and their flowers, based on whether they're monocots or whether they're dicots. So you are going to need to be able to compare the two of them, tell the difference between the two of them as we go through all of this. Now, one thing that I want to point out about the pictures on the bottom of page 483 that are pictures of monocot and dicot roots, I'm not sure why they did this. I don't know if it's because they ran out of room. They have a whole lot of stuff labeled on the monocot root that they don't have labeled on the dicot root. But it is not the case that these things that they have labeled only exist in the monocot but not the dicot. They actually exist in both. So in the rather primitive picture that I have drawn up here behind me, I'm going to show you that all of these structures, with the exception of one, are in both the monocot and the dicot root. So the first main difference between monocots and dicots is that monocots have a fibrous root system, which means you pull the plant up and the roots are just going everywhere, whereas the dicots are going to have a taproot system, which means you have just one main root that goes down, as opposed to having roots that kind of go all over the place. And again, there are pictures in your book that can show you what that looks like. You will notice that when I take a cross section of both, they both have an epidermis. Obviously, that's going to be made of your dermal tissue that we talked about. It's there for protection, and because we're talking about roots, this is also the tissue that is going to be responsible for absorbing the water and the minerals from the surrounding soil. Okay, so epidermis on the outside of both of these. Now, while I have this drawn as a line, Remember that in actuality, what this is this is a whole bunch of cells. They're epidermal cells going around. It's not like this, it's not like a ribbon that goes around it. It's not like a string. That's the easiest way to draw it. But if I drew it accurately, I would actually have to draw a whole bunch of little circles so to show you that they're all made up of individual cells. So even though we draw it as a line, keep in mind that we're actually talking about individual cells in each one of these layers. Okay, so we start with the epidermis, and again, that's there for protection. The cortex is next. It's right inside the epidermis, and it's a layer that's bigger in one root than it is in the other. The cortex is basically there for storage. If it's going to store potentially starches and things like that that are going to come down from the leaf. So that's there for storage. The next layer in, and this is where I kind of tried to color code things, the next layer in is the endodermis. And even though it says dermis, it's not made up of dermal tissue. It's actually a different kind of tissue. And this endodermis is, I like to call it a gatekeeper, because what it does is it either allows things to get into the vascular chamber where the xylem and phloem are, because remember the xylem and phloem are going to take things all throughout the plant, and so it either lets things in or it doesn't let things in. Okay, so this endodermis is gonna be a gatekeeper that decides what gets into the vascular chamber and what does not. Now the vascular chamber is everything that's going to include both your xylem and your phloem, and this is one of the things that in the picture on page 483, they have a vascular chamber labeled on the monocot but not the dicot, and there is a vascular chamber in both because they both have xylem and phloem in them. So the endodermis is going to decide what gets into that vascular chamber. The pericycle is the layer of cells right inside the endodermis. Again, remember, these are, even though they're drawn as a line, it's like a ring of cells that goes around there. The pericycle is actually where, if the root needs to branch out, that's where that starts, is in the pericycle. And so if the root comes down and it's going to send a root out sideways from it, the pericycle is the type of tissue that's going to come from. It gives rise to both vascular and the cork cambium in dicots, which we'll talk about a little bit later. So that pericycle is a region for growth. Inside of that, I've got the xylem and the phloem. Now, in a monocot, your xylem and phloem are in rings going around this direction. And of course, the xylem is on the inside and the phloem is on the outside. The xylem in my drawing is represented in blue and the phloem is represented in green. Now, in a dicot root, they're not organized this way. They're organized where your xylem is in an X shape and the phloem is in between the arms of the X. Okay, and again, you can probably see that a little bit better in the blown up picture in your book on the bottom of page 283, and they have it blown up there on the side, so you can see that X shape that the xylem have made. In a dicot root, in between your xylem and your phloem, you actually have vascular cambium in between those two, and vascular cambium is there to make more xylem and more phloem, and it's undifferentiated while it's in the cambium, but then if it needs more phloem, it'll differentiate into a phloem. If it needs more xylem, it'll differentiate that way. Okay, so that is the growth tissue that's in between there, and again, I've got that as kind of little black circles, black layer in between the xylem and the phloem in the dicots. Now, there is no vascular cambium in the monocots, and the reason for that is because the growth in a monocot is much more limited. And so because the growth is limited, the plants just aren't going to get as big. In your dicots, which is going to be a lot of your huge trees, some of your other ones as well, but a lot of your really big trees, they have to be able to keep growing and growing and growing and getting bigger. And so that vascular cambium is necessary so they can keep making more xylem and more phloem so that it's able to keep the tree nourished. Now, the vast majority of your large trees, there are a couple of exceptions, but the vast majority of your large trees are dicots with the taproot system. And I know when I first heard that, I was really confused, because I know when we go walking in the woods around here, we have to be really careful not to trip over tree roots. And I'm like, there's tree roots growing everywhere. How can that not be a fibrous root system? And so I wanted to mention that so that you didn't get confused. In a lot of trees, when the tree first starts growing, the taproot is the main primary root. And then as the tree grows later on, it will develop a lot of lateral roots that are going to come out from that taproot. If we're working out in our garden, we have a woods next to our property, and so it likes to drop all kinds of acorns and maple leaves and all that kind of stuff. And so if a maple tree starts growing in the middle of the garden, my husband yanks it out. You can see that it does have one nice long root on it, but it doesn't stay that way because it ends up having to grow so many lateral roots off of it in order to anchor such a large plant. Okay, so I didn't want that to confuse you. All right, so I think, oh no, then we have the pith. There is pith in the middle of a monocot. There might be a little tiny pith in the middle of a dicot. In a lot of dicots, there isn't any pith at all. And so that's going to be the main difference between your monocot and your dicot roots. It's going to be the arrangement of the xylem and the phloem. They're arranged in completely different arrangements. Your dicots are going to have vascular cambium, whereas your monocot is not. And your monocot is going to have pith in the middle, where your dicot is not. Okay, you're going to be doing a lab a little bit later on where you're going to be looking at slides under the microscope of these different kinds of roots, and you're going to want to be able to identify all the different kinds of tissue. So that's why I'm going to be going through this in detail to help you know what you're looking for so that you can recognize these things when you get to your lab a little bit later.